So this will be the first English presentation. I'm better in English than in Swedish. So my name is Wouter van der Wijngaart. I'm from the Microsystem Technology Group in KTH, or MEMS, if that says anything to you. I'm responsible for fluidics, and my research domain can be maybe best defined as micro, nano, biofluidic systems. I try to go a little bit more in detail. Today I'm only going to talk about lab-on-chip technologies. What we do is system technology, and we go all the way from micro nanoscale physics to fabrication, material development, machining, integration, all the way towards applications, lab-on-chip, and then we typically work together with people with a more medical background to really get something that is also relevant. So what is lab-on-chip? Basically what you would like to do is take a typical lab and integrate everything on a little cartridge, a little chip. So you add your sample in, and on the outside you get in an electronic signal or an optical signal exactly the measurement results you want to have. And we have done lab-on-chip systems for virus detection, protein detection, bacteria, DNA, molecule detection. I'm going to go a little bit more deeper in that. To make such systems, you need a multi- or interdisciplinary team. Typically, you need somebody to make the hardware. You need somebody to understand the bioware, and with that I mean the biochemistry on the chip. And of course, you need somebody with a medical or clinical expertise to tell you what is actually needed, what is there. And then you need somebody to integrate everything onto the single chip. Now, my specialty is this integration part, and I work very close together with 2-3 people from different groups at KTH that could unfortunately not be here within biochemistry and within polymer science. So I can be also linked towards them. So within KTH, we are pretty good in this part. And what I would like to fish up here is this part. Yeah? What do we do on such a lab-on-chip, typically? Well, you add your sample. Typically, you need some sample preparation. We work with blood sample, with airborne sample. So you need to collect, purify your sample. Then you need to do some bioassay, and it can be, for example, PCR, or it can be direct immunobinding or fluorescence or whatever it is. And then also you need, of course, a transducer, because you need to integrate on your chip the readout also. That can be a photonic transducer or a QCM we work with, or electrochemical. And all of that you also need, of course, your sample needs to be pumped through, controlled, valved, this kind of technologies. Hopefully you can do that as simple as possible, but sometimes that's not possible. These are some of the examples of the 5 lab-on-chip diagnostic technologies we have been developing during the past 5 years. Due to lack of time, I only want to touch on 2 of them because I have heard people talk about this kind of problematic, so I want to go a little bit deeper into that. And one is on bacteria detection and typically sepsis. So we are leading a European network with 7 partners in which we try to identify sepsis and antibiotics resistance from a whole blood sample within 1 hour. So what we do is we don't grow the bacteria, but we take 3 milliliters of blood and we try to filter out the bacteria by getting rid of all the other blood cells and up concentration. Once you do that, you do lysis of the bacteria, you do an on-chip PCR, and then you flush you flush it over a photonics readout array, and then you read out whether you have a probe binding or not. So that is typically what we try to integrate on 1 cartridge. Another example is viral detection, and specifically here we look at Wijngaart. We work together with Hans Wiksel and with Lennart, names escape me, from Linköping is Lennart. And what we do there is we make a little chip that collects particles from the air electrostatically, and you capture that on a 1 square centimeter chip, and there's a very little liquid layer there, so you absorb your virus or any particles, and you try to bind that with an antigen-antibody binding directly onto a gold surface on top of a quartz crystal. And you can measure a very small mass change. You can directly measure, do I have this sample there or not? So these are just 2 examples of what we are trying to do. This is underlying technologies that we use. This is not so interesting. This is really more the hardware part of what our group develops. And so what I suggest happens in the next step, if you think that this type of technology is of interest for you, please step up to me, and I would like to set up a more detailed meeting with you, maybe a 2-hour meeting together with my colleagues that know more about the biochemistry, more about the materials. But because alone I'm not competent enough to answer your medical questions. I'm a hardware guy with an electronics background. So please step up to me. We do lab-on-chip, but we do also other medical technology projects. Okay, I think I stop here.